Hello everyone, happy morning, a very good morning. I hope and I believe all of you are doing well. Am I audible and visible? Yes, am I audible and visible to everyone? Yes, that's great. So today in this session in our NEET PG Rank Booster Revision series that we have started since last 3 days, we are going to discuss the very important and confusing topic of bacterial toxins, the mechanism of action, which acts by inhibiting protein synthesis, which acts by increasing cyclic AMP, or which acts by increasing cyclic GMP. Yes, Rose, this class will be available later on as well. Alright. So now to begin with the class at the outset, can you tell me which is that bacterial toxin which acts by increasing cyclic GMP? Which one acts by increasing cyclic GMP? Hearing test, Arzu, I've already taken on the Unacademy Plus platform. So if you are an Unacademy Plus subscriber, you should be able to see those classes. Very good, amazing, Shiva Pranil. That is your heat stable toxin of E-Tech, which is your cyclic GMP. Absolutely right. And the heat labile acts by increasing the cyclic AMP. So let's decode this topic of bacterial toxins. I hope all of you have watched the previous 3 sessions in your NEET PG Rank Booster Revision series. 2 we had an ophthalmology, 1 was on your INO, 1 was on your what was the second topic that we had in ophthalm? Predominant astigmatism was very important. Then yesterday your nerve fiber classification. Then we had a quiz also. I've shared the notes also, my own handwritten notes on the Telegram group. And today also I'll be sharing, if you want, I'll be sharing these notes of bacterial toxins mechanism of action. So you can just focus and listen very attentively while attending this session, and then I can share these notes on the Telegram group. Okay. So now starting with the topic of bacterial toxins. First we have exotoxin and we have endotoxin. The trick to remember is that your endotoxin is present in gram positive or gram negative. Endotoxin is present in gram positive or gram negative. So endotoxin, remember endo is N, which is your gram negative. Okay, endo is N. It is present in your gram negative toxins. The 1 gram positive bacteria which is having endotoxin. Gram positive which has endotoxin is, gram positive with endotoxin is Listeria, right? It is your Listeria. Then Listeria is what we have read is important for your neonatal meningitis as well. During the delivery, it can cause your infection to the newborn and all those things we have seen. Abortions ke liye it is one of the predominant cause. So exotoxin versus endotoxin, as the term says, exotoxin, exo matlab it is secreted out of the bacteria and then it acts. While your endotoxin, endo is within the, it is the structural component, okay, it is the structural component of the gram negative bacteria. So a very important question, where is the endotoxin present? Is it present in the cell wall? Is it present in the outer cell membrane, the inner cell membrane? Where is it present? Where is the endotoxin present? Is it cell wall, outer cell membrane, inner cell membrane? Where is it present? No. Remember, it is present in your outer cell membrane. It is your outer cell membrane. Now, your endotoxin is made up structurally of It has a O antigen, it has a core polysaccharide, and it has your lipid A. Okay, lipid A. These are the areas I'm telling you which you might have missed while reading but can be asked in your exam. Out of these, which is the toxigenic component which is responsible for the toxin wala action? Is it all of the above? Is it O antigen, the core polysaccharide, the lipid A, or all of these? Please remember, it is the lipid A. Okay, it is the lipid A which is the toxigenic component. Okay, that is the toxigenic component of the endotoxin. It is basically in your lipopolysaccharide. It's a lipopolysaccharide, lipid and polysaccharide both. It's the lipid component which is the toxigenic component. Okay. And this endotoxin, generally they are present in gram negative, like your Neisseria, we know meningococcemia, it causes your DIC and all. So they cause your increased cytokines release, like your interleukin 1, TNF alpha and all of those. And they cause your shock and all can come with your endotoxins. Okay, shock and all can come with endotoxins. Which is the gram negative organism which has your exotoxin as well? Gram negative which has exotoxin. Its mechanism of action is by inhibiting protein synthesis. It inhibits your protein synthesis by ADP ribosylation of elongation factor 2. Which is a gram negative organism with exotoxin which inhibits protein synthesis. Why? I've given you all the clues to answer this question. Absolutely right. That is your Pseudomonas. Remember, Pseudomonas has something called as exotoxin A. Okay, it has your exotoxin A as well. And I asked gram negative. Corynebacterium is your gram positive, right? It's your gram positive. Which is more antigenic? Which is more antigenic or has more adverse effects? Is it exotoxin or endotoxin? Remember, it is your exotoxin. Okay, it's your exotoxin which is more antigenic. Excuse me. Right. So your toxoids and all can be prepared from your exotoxin. Alright. Let us see this table now. The table which shows the difference between exotoxin and endotoxin. So exotoxin is your certain species of gram positive and also your gram negative. Endotoxins is present in your outer cell membrane of most gram negative bacteria. Gram positive is Listeria. Exotoxin is secreted. This is not secreted. Exotoxins are generally polypeptide. Endotoxin, it is the lipid A component of the lipopolysaccharide, which is the structural part of bacteria. It is released when lysed. The gene for exotoxin is your plasmid or bacteriophage. We know that Corynebacterium diphtheria. कौन सा फेज होता है बैक्टीरियोफेज? प्रोफेज, अल्फा, बीटा, गामा, डेल्टा. कौन सा प्रोफेज का रहता है which gets the toxin basically? Is it your alpha, beta, gamma? What is it? Please remember, it is your beta prophage, right? It is your beta prophage. So your Corynebacterium diphtheria, it is active. The toxin is active by bacteriophage. That is your lysogenic conversion, right? The attack of bacteriophage, that is your lysogenic conversion. What is that molecular biology में technique में हमने पढ़ा? Bacteria has at that inherent immunity to bacteriophage. What system do they use to protect themselves from the bacteriophage? What is that system? Which we use in your molecular biology for treatment, for inducing some genes. Yes, that is your CRISPR, right? That's your CRISPR. CRISPR is used to protect from the bacteriophage. While your endotoxin is inherent structural है, तो वो bacteria के chromosome में ही होता है. It is in the bacterial chromosome. Adverse effects, it is high. These are fatal in the order of 1 microgram. This is low. Fatal dose is more. Clinical effects, you'll see various effects and all. This is fever, shock, DIC, like you see with meningococcemia, like your Waterhouse-Friderichsen syndrome. It gram-negative endotoxin, it induces TNF, interleukin 1 and interleukin 6. Antigenicity, this induces high antibodies, that is antitoxin. So toxoids are used as vaccines. These are poorly antigenic. No toxoids, no vaccines available. Heat stability, endotoxins are heat stable generally. Exotoxins are heat labile. Except which is the heat stable exotoxin? Remember staphylococcal ka enterotoxin and your E. coli heat stable toxin. These are heat stable, otherwise exotoxin are heat labile. So tetanus, botulism, all of these are your exotoxins. Okay. Now coming to your mechanism of action of the various toxins, the bacteria with toxins, what is the mechanism of action of various toxins? Okay. So this we have seen basically, जो हमने अभी पढ़ा, the exotoxin and endotoxin, heat stable and all of those, the location of genes. Coming to exotoxin. The first mechanism of action of exotoxins is, by inhibiting your protein synthesis. Okay, it is by inhibiting your protein synthesis. Now, which are the organisms which inhibit your protein synthesis? By ADP ribosylation of elongation factor 2. Remember, the mnemonic is CDEF, that is Corynebacterium diphtheria, acts by ADP ribosylation of elongation factor 2. Also your Pseudomonas ka exotoxin A, that also acts by elongation factor 2. So you can remember this as elongation factor 2 is di or do. That is your Corynebacterium diphtheria and it is your Pseudomonas. So elongation factor 2 is di and do. Okay. The next bacteria which inhibit. Is it stuck for anyone? I hope this is fine with everyone. Right? Okay. The next bacteria which act by inhibiting your protein synthesis but by inhibiting 60S ribosome. They inhibit your 60S ribosome. The mnemonic for that is 60, pronounce it as 60. So you know that it is Shigella and the Shiga-like toxin, that is your Verotoxin. These act by inhibiting 60S ribosome. This Shiga-like toxin is present in your enterohemorrhagic E. coli, right? Your enterohemorrhagic, which basically cause your hemolytic uremic syndrome, HH, hemorrhagic hemolytic uremic syndrome. The difference is Shigella invades the GI mucosa and that is why it causes your dysentery. This causes your dysentery. While your Shiga-like toxin, enterohemorrhagic, does not invade the mucosa. Okay, it does not invade the mucosa. Right? So clear with everyone which are the bacteria which inhibit your protein synthesis? Which ones inhibit your protein synthesis? So 2 acting by elongation factor 2, 2 acting by inhibiting 60S. Elongation factor 2 is Corynebacterium diphtheria and your Pseudomonas exotoxin A. 60X is your Shigella and the Shiga-like toxin of enterohemorrhagic coli, that is also called Verotoxin, causes your hemolytic uremic syndrome. Next one, the next mechanism of action is by increasing the fluid secretion. So we know that for fluid secretion it is your cyclic AMP, cyclic GMP. Cyclic AMP increase करने वाले 3 हैं, cyclic GMP increase करने वाले there is only 1. So the mnemonic to remember is labile in the air and stable on the ground. That means the heat labile toxin of ETAC, labile in the air is your cyclic AMP. Stable is on the ground, that is your cyclic GMP. So remember, labile in the air, that is labile by cyclic AMP. Stable is by ground pe, that is cyclic GMP. Now the next ones which act by your cyclic AMP is your anthrax and it is your cholera. Okay, anthrax and cholera. There's one more toxin which also acts by increasing your cyclic AMP. Can anyone tell me which is that? Which is that toxin which acts by your increasing cyclic AMP apart from all of this? Anyone? Yes, Himani, this class will be available for later viewing as well. Absolutely right. That is your pertussis. Okay, that is your pertussis, which also acts by increasing your cyclic AMP. So anthrax, may have written this as edema factor. So you know that anthrax, your malignant pustule, the cutaneous anthrax, there is surrounding edema. That surrounding edema is what is your edema factor. Remember, it is basically edema is because of increased cyclic AMP. So the trick to remember your cyclic AMP, I think we have it on the last page here. Yeah. So which act by increasing cyclic AMP? Remember, the trick is cyclic AMP. Cholera, anthrax, this M is, you make it like horizontal, that becomes E. So your E tech labile in the air, E tech labile, and P for pertussis. So remember, the toxins which act by increasing cyclic AMP are your same CAMP, cyclic AMP. Cholera, anthrax, M is like E, that is E tech labile wala, and pertussis. E tech stable is by cyclic GMP. Okay. Now these are your cyclic AMP and cyclic GMP. Now phagocytic ability, if you get a question on that. So remember, that is also pertussis. Okay, that is also pertussis. So remember, pertussis inhibits your phagocytosis and it prohibits your GI. Okay, it prohibits your GI subunit. So you have GS and you have GI, G stimulatory protein and G inhibitory protein. When you activate your G stimulatory, it increases your cyclic AMP. Let me write down here. Okay. So you have GS and you have GI. When you stimulate your GS, it increases your cyclic AMP. When you inhibit your GI, it increases your cyclic AMP. So the toxin which acts by inhibiting the GI, not by stimulating the GS, it is basically inhibiting is prohibiting. So remember, it is your pertussis. While cholera wala acts by, it is GS, it stimulates the GS. These are acting basically by your ADP ribosylation. ADP ribosylation of GS is cholera, GI is your pertussis. Okay, GI is pertussis. Next one, so this is what we said, cholera increases cyclic AMP by stimulating the GS. While pertussis prohibiting, it increases the cyclic AMP by inhibiting your GI. Next mechanism of action, neurotransmitter release inhibition. Whether it is inhibiting your inhibitory neurotransmitter or excitatory. Inhibitory neurotransmitter is your glycine and GABA. If you inhibit the inhibitory neurotransmitter, that means it's all excitatory, it is spastic paralysis. We know your spastic paralysis is seen with tetanus, so Clostridium tetani. When you inhibit your excitatory, it is your flaccid paralysis. Flaccid paralysis like floppy baby is seen with botulism. Remember, floppy baby, very, very important. When you give honey, important for botulism is canned food. So you can remember this as canned food is basically the food kept in bottle. So whenever you get this history of a person eating something from canned food and then getting flaccid paralysis, that is botulism. So can is like bottle. So canned food is botulism. Honey, floppy baby is botulism. Now these act by inhibiting your snare. What is the snare? Snare is the receptor, the protein which is required for the release of neurotransmitter. So you can, this question has also been asked, which of the following toxin acts by inhibiting snare? So remember, basically this is your neurotransmitter release. Okay, it is your neurotransmitter release. Snare is required for that. You can remember that. It is neurotransmitter release, though the full form is something else. Alright. Next one. What do we have? The next one is the last category in exotoxin, the fifth one, the bacteria which act by lysing the cell membrane. They lyse the cell membrane. Now which one lyse the cell membrane is which ones lyse the cell membrane? Clostridium perfringens and Streptococcus pyogenes. You can better remember this as When you say phospholipase or lecithinase, we know that it's present in Clostridium perfringens. Which reaction it is? Your Naegler's reaction. So they break the phospholipid. That means they are lysing the cell membrane. It is your alpha toxin. Very important. Again, is it your alpha, beta, theta toxin? It is your alpha toxin of Clostridium perfringens, because that is what causes your Naegler's reaction. So it causes the cell membrane lysis. So it leads to your myonecrosis, that is gas gangrene. Lysis of RBC membrane leads to hemolysis. What do you get on hemolysis? Double zone, like your target hemolysis. The double zone hemolysis is seen with Clostridium perfringens. Please remember that. Next, your streptolysin O. As the term says, lysin, it lyses the cell membrane of Streptococcus pyogenes, not Streptococcus pneumoniae. It lyses the cell membrane, and that is how it causes your beta hemolysis. Your anti-streptolysin O titers we use for diagnosis of rheumatic fever, right? Streptococcus common cause. So ASO titers are used for diagnosis of rheumatic fever. So the next category in your mechanism of action is your cell membrane lysis. Lysis, you know, is phospholipase or lecithinase. That is your Clostridium perfringens alpha toxin. It gives your double zone of hemolysis. Streptolysin O, okay, it is lysing. It is your Streptococcus pyogenes. It is used for diagnosis. Anti-ASO titers are used for your diagnosis of rheumatic fever, right? Diagnosis of rheumatic fever. So everyone clear? Very good, Tanmay. That's a good mnemonic there. Perfringens is perforate. Okay, perfringens is perforate. Okay, next. So these are the five mechanism of action that we have seen. One is your protein synthesis, then is your fluid secretion, you have your neurotransmitter release, phagocytosis inhibition, and you have your cell membrane lysis. Okay, and you have your cell membrane lysis. So that was about exotoxins. Now coming to the next one, we will revise this quickly later on. Super antigens causing the shock. Okay, super antigens. Which are the super antigens? Staph aureus ka TSST, the toxic shock syndrome. Now super antigens causing shock, so they cause your sepsis, they cause fever and shock. So your toxic shock syndrome, it itself says it's causing shock. It's your super antigen. Why are they super antigen? Like it's causing your polyclonal T cell activation. A lot of T cells are activated outside the antigen binding site, and it leads to lot of your cytokine release that leads to shock. Okay, and also your Streptococcus pyogenes. Which toxin is super antigen? It is your erythrogenic exotoxin A, which causes your scarlet fever. Okay, which causes your scarlet fever. Some important points about scarlet fever. S for S, sandpaper rash. It is also called second disease. You have circumoral pallor, an important clue in the question. You have Pastia's lines in the fold, very important. And there is strawberry tongue. First, it is your white strawberry tongue, and then it turns into the red color. So white and red. So remember, sandpaper rash for scarlet fever, second disease, strawberry tongue, circumoral pallor, and your Pastia lines. These are the important points. What is the mechanism of action of these super antigens? Basically, they cross-link your T cell receptor to the MHC2 on the antigen presenting cells outside the antigen binding site. Basically, there is increased cytokine release that leads to shock. So remember TSST and your erythrogenic exotoxin A. Which are the other toxins of Staph aureus? You have your exfoliative, as the term says, exfoliation karne wala, which causes your scalded skin syndrome. So if you get a question, scalded skin syndrome is caused by exfoliative toxin, TSST, or heat stable toxin. It is caused by exfoliative toxin. Heat stable enterotoxin. We said that staph is a exotoxin, which is heat stable, the enterotoxin. The other one which was heat stable was, one was staph and the other was heat stable, your E. coli, the E-tech wala, which acts by cyclic GMP. That causes your food poisoning, which classically they give you that within 6 hours the patient has the symptoms, because it's a preformed toxin, it acts very fast. So within 6 hours your Staph aureus manifests food poisoning. Coming to your endotoxin, which is present generally in gram-negative, gram-positive listeria, composed of O antigen, core polysaccharide, lipid A. Lipid A is the one which is your toxic component. When is it released? Upon the cell lysis. What are the three main effects? Your endotoxins. They cause macrophage activation, they cause complement activation, they cause tissue factor activation. Important point to remember here for macrophage, right? For macrophage, which is the CD marker? How do we remember that? For macrophage, the CD marker is, this is 1 and this is 4. It is your CD14, which is basically the receptor for your LPS, lipopolysaccharide. Very, very important question asked in your exam. Which is the CD marker for your lipopolysaccharide and which is the toll-like receptor? 1, 2, 3, 4, 5, 6, 7, 8. It is your TLR4. Remember CD14 and TLR4. So your endotoxin acts on the CD14, which is like your TLR4, and it activates your macrophages. So remember TLR4 and CD14. Okay, is this clear with everyone? Yes, so macrophage is CD14 and TLR4. Very, very important. The previous NEET PG has a question on this, which is the toll-like receptor for LPS. It is your TLR4. Very, very frequently asked. So let us quickly revise cyclic AMP. We saw the mnemonic. The mnemonic itself is cyclic AMP. Your cholera, anthrax, M को E जैसा देखो. So your E tag का air, labile in the air, and your pertussis. The difference is pertussis is prohibiting, it is inhibiting GI. Cholera wala, it stimulates your GS. These act by ADP ribosylation. E-tech mein you have heat labile in the air, cyclic AMP, heat stable on the ground, cyclic GMP. Toxins acting by ADP ribosylation of EF2 and your GS and GI. Already we saw EF2 is diphtheria and pseudomonas, and your GS is your cholera, also your labile wala, which increases the cyclic AMP. Pertussis is your GI alpha subunit. Remember, pertussis is prohibiting GI. It inhibits your phagocytosis as well. Important point, remember that the toxin A and B of Clostridium difficile, these are glucosyl transferase. Okay, these are glucosyl transferase. Clostridium difficile ke toxin are glucosyl transferase. And diphtheria toxin, we said it is active by your lysogenic conversion. Beta prophage is required, right? It's your beta prophage which is required. So that was about the toxins ka mechanism of action. I hope all of you are clear on this, right? So let us see this table. Protein synthesis. Just a second. Okay. So protein synthesis, diphtheria, Pseudomonas, Shigella, EHEC. This is your EF2, di and do, Shigella and EHEC, Shiga-like toxin. It is your 60S ribosome. Second one, fluid secretion increase karne wale, cyclic AMP, cyclic GMP. Stable is ground. It is your cyclic GMP. Rest are your cyclic AMP. Then we have your phagocytosis. Pertussis prohibits your GI. It causes your cyclic AMP increase. Then you have Clostridium tetani and botulism, neurotransmitter release, that is inhibiting the snare. Inhibitory inhibit karne wala, lockjaw tetanus, and stimulatory inhibit karne wala, that is flaccid, is botulism. Lysing cell membrane, perfringens perforates, and your pyogenes perforates. You can remember both of them, PP. Alpha toxin and streptolysin O, right? Alpha toxin and streptolysin O. So you have your double zone hemolysis. Here you have beta hemolysis. That is what is important. Super antigens, toxic shock syndrome, erythrogenic exotoxin A, and it causes your scarlet fever, toxic shock-like syndrome. Okay, toxic shock-like syndrome. Then you have your endotoxins, LPS present in outer membrane. The lipid A is the toxic component, right? And it is your TLR4 CD14, which is macrophage activation. Okay. So the final summary वापस कर लेते हैं. Cyclic AMP increase करने वाले हमने देखा cholera, anthrax, this is ETAC, heat labile, and your pertussis. So remember, this is pertussis, anthrax, cholera, and ETAC का heat labile toxin, labile in the air. Cyclic GMP, stable on the ground. Protein synthesis, EF2 is di, diphtheria and pseudomonas. You have your difficile also, which is di. Remember, those are your glucosyl transferase. Then you have 60S, Shiga, that is your Shiga-like toxin, EHEC, and Shigella dysentery. These inhibit your 60S, right? These inhibit your 60S. Clear with everyone? Right? Yes, I'll share these notes, guys. You can quickly revise after this session itself. Today itself, revise these notes so that it's easy for you to remember these toxins ka mechanism of action. On this page, you have the summary of all the toxins jo humne dekhe, exotoxins, protein synthesis, fluid secretion, phagocytosis, cell membrane, neurotransmitter. So in one page for exotoxins, this has been summarized. So you can definitely revise these, and I hope this will help you. And thank you so much for joining in. I hope all of you remember. When are we meeting next? We are meeting next at 5 p.m. Okay, we are meeting next at 5 p.m. Since our daily target is physiology, so some high yield rapid last minute revision points from physiology we will try and cover in the today's 5 p.m. class. Some concepts there, your renal physiology ke formulae, some important graphs, whatever is important I'll try and cover in the today's 5 p.m. class. And I'll try and arrange a quiz as well based on the clinical questions for your toxins mechanism of action. So like we had a quiz yesterday on nerve fiber classification, I'll try and post a quiz on your mechanism of action of bacterial toxins, integrated clinical questions, not like there would be some one-liners and there would be some clinical questions. Okay, so stay connected on Telegram group, stay connected on Instagram, and MCQ के साथ, are you talking the word details? Are you talking about the physiology class? Yes. So we will get some MCQs as well. Don't worry on that. And quiz after 10. Yes, Aman. I'll maybe reschedule the quiz timings. But abhi jab now 9 o'clock it's going on. So many students are participating. Tab tak karte hain. Tomorrow 5 p.m. I'll give you an update on that, whether we have physiology itself or maybe there would be biochemistry, radiology integrated class. I'll give you an update on that on Telegram group. Okay. So thank you so much everyone for joining in. Do subscribe to this YouTube channel so that you know whenever there are new sessions, there are new videos or anything new coming up, you get a notification for that. You do not miss on anything. And please let your friends know about these sessions so that more students can join in. And thank you for all the love. See you at 5 p.m. on Unacademy app for a free live class. Till then, goodbye, take care, keep studying, keep revising and keep winning.